Hello, everybody. I would like to present a research under a NASA interdisciplinary science grant to assess soil-borne plant pathogen dispersal across global dust currents by building a remote sensing system for plant disease. This work is conducted by an interdisciplinary team, which is led by me as the lead postdoc of the project, together with the collaborators from Cornell University, Penn State University, and the JPL. First of all, why bother for moving towards global disease surveillance using remote sensing? The reality of plant disease in the world is that it causes from 15 to 60% yield loss annually, which turns into $220 billion loss every year. Additionally, plant diseases cause biodiversity loss due to the spread of invasive plant pathogens, the appearance of new virulent strains, and the overuse of pesticides. Focusing on soil-borne pathogens, in particular Fusarium oxysporum, which is the target of our project, it is endemic to the 6 continents and has more than 100 susceptible hosts. In addition, it can survive in soil for more than 20 years, removing land from agricultural production, and its ranges are expected to expand under predicted climate change scenarios. Because of the grand scale of these problems regarding plant disease, a group of world pathologists cashed up together in 2019 to offer a wonderful science paper about the need for a global surveillance system for crop diseases. But also the use of remote sensing offers opportunity to detect and predict pathogen dispersal. Despite this paper had no mention about the role that remote sensing could play on it. This shows how distant the field of plant pathology and remote sensing are right now. Our project bridged this gap by laying the foundation for a global remote sensing-based crop disease surveillance system for Fusarium wilt, taking into account previous studies that demonstrated that soil, dwelling fungi are capable of aerosolization and transport in global dust plumes, and that infectious Fusarium oxysporum spores and DNA have been isolated from North African and Asian dust samples. It inspired ideas for this project. We wanted to combine these 3 separate disciplines into building a global system for surveillance and monitoring Fusarium oxysporum by determining how remote sensing measurements and Earth system models used to track atmospheric aerosols can be informed by comparative genomics to improve food security. Remote sensing could map disease distribution, informing where the incident is. Aerosol transport modeling could map dispersal, informing where the dispersal risk is. And comparative genomics is able to assess how likely spores from source and sink regions are related, informing about pathogenicity and spore variability. The project structure sets that for the first year, each discipline has independent goals, and for the following 2 years, the goals tend to merge together. The goal for the first year of project regarding remote sensing is to build a susceptibility assessment for current Fusarium oxysporum risk in agricultural zones from remote sensing measurements. Aerosol transport goal is to adapt an existing model to include agricultural dust and spore transport. And comparative genomics goal is to assemble spore traits that impact dispersal and atmospheric viability. Jumping into the susceptibility assessment, we are using the framework of plant disease triangle in order to map disease distribution. The plant disease triangle is the foundation of theory that plant disease is the product of a virulent pathogen interacting as a susceptible host within a conducive environment. And only when you have these 3 angles together, you will have plant disease. We especially restricted our assessment to areas with known Fusarium wilt, building and uploading a global incidence geodatabase online through a web map. To map conducive plant disease environment, we use Fusarium oxysporum distribution from web map and satellite data products such as SMAP, GPM, MODIS, and EcoStress within species distribution models. Finally, these maps were overlapped with agricultural production zones using the recent wall cover product from the European Space Agency. The goals for the atmospheric modeling side were to compare the existing Earth system model CAM6-MIMI to existing observations by parameterizing the model for the dust event Godzilla in summer 2020. Additionally, this model was modified to incorporate agricultural dust which is our target here because we want to know if agricultural dust is carrying spores of Fusarium oxysporum. The next steps here are incorporating Fusarium oxysporum spores into the model and conducting a survival assessment of a spore by killing them during transport. These tasks are informed by comparative genomics findings, which assemble spore traits that influence dispersal and atmospheric survival, such as size, shape, and ranges of temperature, humidity, pressure under which they are able to live. The significance and impact of this project is, first of all, that this work will build the foundation to assess the dispersal of soil-bound spore on global dust currents as a new element to the Fusarium wilt cycle. Also, it will increase our knowledge of the capacity of remote sensing and earth system modeling to detect, quantify, and inform risk of biotic stress in agroecosystem at global level. Thus, by providing this tool to preserve existing agroecosystems, we will be able to conserve biodiversity and natural ecosystems. Finally, this work will lay the foundation for future use of SBG, imaging spectrometer, integrated with other remote sensing tools as a global surveillance system for crop disease. Thank you very much. Please contact me if you have any questions.